Guys, today I'm going to talk about trapping light energy. Okay, so let's get started. Chloroplast contains several different pigments, and these different pigments absorb different wavelengths of light. The photosynthetic pigments of higher plants form two groups, the chlorophyll and the carotenoids. And chlorophylls absorb mainly in red and blue-violet regions of the light spectrum. They reflect green light, which is why plants look green. Chlorophylls absorb mainly in red and blue-violet regions of the light spectrum. They reflect green light, which is why plants look green. The structure of chlorophyll A is shown in here, and carotenoids absorb mainly in blue-violet region of the spectrum. An absorption spectrum is a graph of the absorbance of different wavelengths of light by a pigment. An action spectrum is a graph of rate of photosynthesis at different wavelengths of light, and this shows the effectiveness of the different wavelengths, which is of course related to their absorption and to their energy content. The shorter the wavelength, the greater the energy it contains. If you illuminate a solution of chlorophyll A or B with ultraviolet light, you will see a red fluorescence, and in the absence of safe ultraviolet light, you can illuminate the pigment with a standard fluorescence tube. So the ultraviolet light is absorbed and electrons are excited, but in a solution that will contain extracted pigment, the absorbed energy cannot usefully be passed on to do work, and the electrons return to their unexcited state, and the absorbed energy is transferred to the surroundings as thermal energy and as light at a longer, less energetic and wavelength than that which was absorbed, and is seen as the red fluorescence in the functioning photosynthetic system. It's this energy that drives the process of photosynthesis. So you can easily extract chloroplast pigment from a leaf to see how many pigments are present by using paper chromatography is shown in here, and you can calculate the RF value for each pigment using this equation. So equation is RF equal to distance traveled by pigment spot divided by distance traveled by solvent. So these will vary depending on the solvent used, but in general carotenoids have RF values close to 1, and chlorophyll B has a much lower RF value, and chlorophyll A has an RF value between those of carotenoids and chlorophyll B. So guys, that's all. Thank you for watching. Don't forget to subscribe and share with your family and friends.